Hello and welcome everybody to today's Drug Hunter Flash Talk. I'm your host, Dennis Koester, and we will be talking about the largest small molecule, the manufacturing process for Enlicitide with David Thaisrivongs today. Good morning, David.
Good morning. Thank you for the invite. Great to be here.
Super cool. Yeah, thanks for coming. And now, without further ado, I want to introduce our guest, David. David Thaisrivongs is currently Executive Director and Head of Biocatalysis at Merck. He obtained his undergraduate degree in biochemical sciences at Harvard University with Dave Evans and his PhD in chemistry at Stanford with Barry Trost. At Merck, David has built and led teams that have advanced pipeline programs from discovery to launch. And highlights of this work include the commercial manufacturing processes for Zepatier, Verubaxstat, Bridion, and nemtabrutinib. Since 2017, he has led the drug substance team for the PCSK9 inhibitor Enlicitide decanoate that we will hear more about today. That's Merck's first macrocyclic peptide being evaluated for the treatment of hypercholesterolemia. And David also serves as a member of the International Consortium for Innovation and Quality in Pharmaceutical Development Drug Substance Leadership Group and is co-founder of the IQ Synthetic Peptides Working Group. In 2022, David was awarded the Early Career Investigator Award by the ACS Division of Organic Chemistry, and he also co-founded the Merck Next Gen Leaders in Chemistry and the ACS Future Pharma Innovators Mentorship Programs. Such an honor to have you here today, David, and what an accomplishment with you and your team to have, manufactured such a massive molecule on scale. So really excited to hear more about it today.
Thank you. It's very, very kind, Dennis.
Awesome. Yeah, and before we get started with your presentation, David, just in the good old Drug Hunter tradition, we want to learn a little bit more about our speakers. So can you share a little bit more about what inspired you to start a career in drug discovery?
So I went into grad school thinking that I would become an academic, like many of grad students. But throughout my graduate career, as I was thinking about the kinds of problems that I would want to apply, what skills and talents I had developed as a scientist, the kind of really significant problems that the biopharmaceutical industry gets to work on, problems whose solutions, save and improve people's lives around the world. Like that was what really drew me to a career in drug discovery and development. So I think you'd find anyone at a biopharmaceutical company in science would echo that kind of real sense of mission and privilege we have in the industry.
Yeah, totally agree with that. Yeah, very probably the most purposeful job you can possibly have. I think I know the answer to that one, but what's your favorite molecule and why, David?
Well, I'm not going to say enlicitide because I'm going to spend the rest of this morning talking about that. But let me point to a different Merck molecule, islatravir, that was recently approved as part of Invinso by the FDA. Uh, I love it because a lot of the work that went into the manufacturing process for islatravir, which we published in Science back in 2019, really laid the groundwork for what I'm going to talk about this morning. So I tell people before the Enlicitide science paper was published recently, that's my favorite Merck paper. If you haven't read the islatravir Merck process, please take an afternoon and take a look at that paper.
Amazing. Yeah, I started my career in virology, so certainly
Yeah.
that's a molecule I'm very much interested in. What are you most excited about in drug discovery today, David?
I love the fact that the kinds of molecules that the industry works on are so much more interesting and varied than they were when I started my career. Like it used to be a very much a small molecule, large molecule dichotomy, but there are all sorts of different sort of weird and interesting molecules that don't neatly fall into either of those two categories. And I think that's an area of really exciting research and also drives a lot of innovation in chemistry and manufacturing to actually meet the challenge of making molecules that the world has not known until very recently.
Yeah, no, totally. Yeah, that's really exciting work that's been coming out of multiple laboratories, especially
Yes.
Merck's. And well, if you could go back to the beginning of your career, David, and give yourself some career advice, or maybe the younger folks in the audience here today, what would you tell your younger self?
It's a great question. I get asked that a lot by students. I think it's important to appreciate that no one's career plays out exactly how they predict. For students out there, you look at someone who you view as really successful. I promise the journey to whatever position they have wasn't exactly how they planned either. And if throughout your career you can be, you know, open to new experiences, like you should be part of organizations that share your values and do work that you find really interesting. But sometimes things don't work out exactly as you plan. And yet if you keep working hard and you're kind and open to new things, amazing things can happen.
Yeah. I like that advice. It's similar to drug discovery. We think it's a linear process, right? But
Far from it, yes.
we take many like rounds and stuff, like many detours to actually arrive at where we need to arrive. But yeah, great advice, David. Last but not least, what do you most like about Drug Hunter?
I love it for the community. I'm here, of course, today to talk about a project. But when I look at people who retire from industry and they write a farewell email to the department, it's pretty rare that they mention, oh, this project that I worked on 20 years ago. It's about the people, the community of science and scientists that we all get to be a part of. That's the best part of the job, and why I love um, places like Drug Hunter that brings the community together to learn from each other, to share science, and to talk about what's next.
Awesome. Yeah, that's very, very kind of you, David. And yeah, so for the people that don't know much about Drug Hunter, I just wanted to quickly share, one feature that we recently launched, which is the molecule search. So here in the molecule search, right on the top left corner, what you can actually do is you can find precedents across the industry for, for example, certain drugs that basically contain specific functional groups. And you can do a search here. For example, if we want to look at the fluoroindole that's contained in Enlicitide, we can quickly draw this out here. And oh, sorry, it actually is substituted as well, but we will put the fluorine here. We'll do a quick structure search. And what we can find is, other compounds that contain this particular motif, and we can see their chemical similarity. We can see whether they're FDA approved. We can see first-time disclosures, modern clinical candidates. And yeah, we can actually play with a plot as well. If we want to see those, for example, alpha disubstituted pyrrolidines that also are contained in Enlicitide, we can do that. We can quickly search here. We can also modify the graph. So if we want to plot, instead of the chemical space, we want to plot the molecular weight against the log P. We can do that. We can already find Enlicitide here with somewhat high molecular weight. David,
kind of like a squiggle drawing of Enlicitide, yes.
Squiggle drawing right there, correct. And yeah, what we can also do is we can even get into like a 3D environment. So if we want to see other molecules again that contain these alpha disubstituted pyrrolidines, and we find one of those interesting, like we can directly go to the PDB, see how they bind to the target. So here, for example, for this particular KRAS inhibitor, with a few clicks, we're right in the PDB structure, and we can see how the molecule binds, what interaction it makes in the pocket, and very quickly learn more about the molecule. So give that a try. We do demos for companies basically daily, and we also can tailor the demo to your specific company. And our customers typically span from single-person biotech founders to the largest pharma companies in the world. And our platform also supports more and more use cases outside of medicinal chemistry. So, for example, in biology, DMPK, even competitive intelligence, medical affairs, or even business development. So if you're interested in a demo of the platform, please let us know after the flash talk or send us an email at info@drughunter.com. And now, without further ado, I'll hand over the stage to David for his talk entitled The Largest Small Molecule the manufacturing process for Enlicitide.
Great. And can I just confirm, Dennis, that this looks good for you?
Looks perfect. Thank you so much, David.
Great, great. Well, thank you again for the opportunity to be here today. And I want to start by saying, I'm one of many, many people at Merck who contributed over the years to this project. So I'm very honored to be here on behalf of a very large and diverse team of scientists and engineers. I want to start this story of the Enlicitide commercial manufacturing process by actually going back to last November. It was a beautiful Saturday afternoon in New Jersey, and I was raking leaves with my two daughters. And in order to bribe them to continue doing yard work with me, we were walking downtown to go to Starbucks to get some sugary drinks from them. And as we were walking downtown, I saw this on my phone. And I have to admit, I told my daughters, Daddy's going to need to use this phone for a little bit. In all of the years that I've been working on this program, I never imagined that I would see the first phase 3 results not only pushed to my iPhone from a New York Times alert, but that alert would be a picture of this beautiful molecule bound to its target. That weekend, we had given the exclusive to the New York Times about the phase 3 results showing substantial reductions in cholesterol in phase 3. And what I love about this New York Times article is that this is the top comment that describes it as a remarkable achievement on behalf of the pharmaceutical industry, and that there are so many people who complain about the cost of medications, but few recognize the investments needed to bring such drugs to market for the patients who need them. And I also love that Malam, if that's their real name, this comment is filled with typos, which to me means it's probably not ChatGPT, or maybe ChatGPT has gotten so good that it now adds in typos. It was really amazing to see over that weekend how this story was picked up through both the sort of trades and the popular press, and that the community really recognized the opportunity Enlicitide has provided to help millions of people manage their cholesterol worldwide. We understand really well how improving cholesterol can significantly reduce cardiovascular events. Cardiovascular disease, as many people know, is the leading cause of death on the planet. And yet there's relatively low patient awareness of this disease. We estimate there are over 80 million people who are treated but not yet at goal for their cholesterol level in the US, Europe, and Japan, which represents an enormous disease burden that we hope molecules like Enlicitide can make meaningful progress. If you look at the patients around the world who are treated for cardiovascular disease, the vast majority of them only are administered statins, and a very small proportion, maybe 1%, benefit from PCSK9 inhibition. Our goal at Merck with the Enlicitide program is to discover, develop, and commercialize the most effective and accessible cholesterol-lowering pill. And we set out to do that by taking advantage of PCSK9 inhibition. It's well known that molecules that inhibit PCSK9 can meaningfully and safely reduce patients' cholesterol. And the reason why we know that is because there are already 3 approved products that target PCSK9 that have this really profound impact. The challenge with these therapies, unfortunately, is that they're all large molecules that are delivered via injection. And what we've seen since their approval is relatively limited commercial uptake. At Merck, we believe that if we were able to translate this breakthrough therapy into an orally bioavailable drug, we would be able to enable broad global access to this powerful mechanism for cholesterol reduction. One of the jokes at Merck for almost a decade now has been whether or not enlicitide and molecules like it are large, small molecules or small, large molecules. And I suppose it depends a bit on where you sit in the organization. Uh, what is unambiguous is that it is much smaller than the large molecule PCSK9 inhibitors that have been approved worldwide. At only 1% the molecular weight, it is a real triumph of molecular design. And, Dennis and I share a heritage with, the Barry Trost group at Stanford. And one of the things that he would always talk about was the importance of atom economy. And if I look at the comparison between enlicitide and a monoclonal antibody, I think Barry would agree. Very atom economical solution to the problem of PCSK9 inhibition. Uh, I often share this slide, and it's not just because I'm giving a drug hunter flash talk. It's been really validating to see the response from the community. And what people might not know is that in addition to sort of the annual Molecule of the Year contest within Drug Hunter also publishes some of the comments that the community provides on these molecules. And so I pulled from Drug Hunter some of the anonymized feedback from voters, and they speak to the kind of work that we at Merck are so excited to be engaged with. And it's also an excuse for me to share a picture of some of the discovery chemistry team at Merck who were awarded a couple of years ago the Edison Patent Award for the enlicitide drug discovery. But I'm here representing the process development organization. So I'm going to move to our work to actually translate this opportunity into real world impact. So enlicitide, you can break up into roughly 10 building blocks. Some of them are amino acids, some of them are non-canonical amino acids, some of them aren't even amino acids at all. And while I'm not a mathematician, I think 10 factorial is over 3 million possible routes, which is a real challenge when it comes to designing the optimal synthetic route. So we have to constrain the design space with a couple important principles for our commercial process. The first is that we want to maximize the convergent assembly of enlicitide from building blocks. We want to control the macrocyclization events. Chemists will appreciate that macrocyclization is often very challenging to do because you're always at the risk of oligomerizing the linear precursors. We want to avoid protecting groups, and we want to avoid any use of chromatography. So while we didn't look at 3.6 million routes, we looked at quite a few. For the purposes of today, I want to narrow down on the one that we selected. Last year, we published our clinical supply process. And while step count is not everything when it comes to synthetic chemistry, I kind of grates on me a bit that most people outside of chemistry know us for our step count and our yield. Step count is not a terrible approximation of efficiency. And what you can see that as we were producing enlicitide, we had reached this asymptote or this plateau of how much more efficient optimizing known chemistry was going to be. Now, this work that was described last year was remarkable. It represented more than a 90% reduction in the cost, but we believe that to actually make this molecule commercially viable, to meet our aspiration of a broadly global accessible therapy, we would need to do substantially better than known chemistry. And if there's one takeaway I'd like to leave you with in terms of the strategy of synthesis, is that while you can make a tremendous amount of progress in optimizing a route by improving a catalyst or reducing solvent usage or improving the yield, if you're optimizing a non-ideal route, ultimately you might fall short. And we knew we needed a breakthrough innovation to enable a better route to enlicitide. Here's the route we wanted to enable. We wanted to take enlicitide and break it into two fragments that we call diamine and western fragment. Western fragment comes from 4 of the building blocks you saw on the previous slide. Diamine is also broken into 2 fragments we called northern and eastern. Eastern comes from 3 of the building blocks and northern from the remaining 3. But this route, as I lay it out simply, is impossible using existing synthetic chemistry methods. We would need a breakthrough innovation to enable this route. Now, I'm part of the biocatalysis group at Merck, and one of the great uh, benefits of using biocatalysis or using tools in biology to solve problems in chemical synthesis is that using biocatalysts enable us to select superior routes that are impossible with chemical reagents. The specificity of enzymes often allows multiple enzymes to work together to rapidly assemble molecular complexity. And modern protein engineering allows us to take natural enzymatic starting points and evolve them to perform unnatural functions. And I'm going to illustrate what this means in the context of the Enlicitide process. But let me give you a very brief overview of what protein engineering looks like. So let's say, for example, you have some starting point in nature which has no overlap with some desired function you have for some unnatural chemical transformation. You might need to progress through protein engineering or directed evolution, enzyme libraries that get you closer and closer to the desired function. And what this often looks like is performing rounds of evolution where you modify the sequence space of your enzyme or your library of enzymes until it gets closer to your desired function. And as you unlock reactivity B, you're able to shift the protein engineering to a new target, and you go around and around this design, make, test cycle until you've achieved your desired enzyme function. So I'm a synthetic chemist by training, so let me make this even simpler for people like me. If, for example, what you have is a cat and what you need is a unicorn, you might first have to transform that cat into a dragon, and from a dragon to a Pegasus, and finally from a Pegasus to a unicorn. Let's talk about that in the context of the synthesis of the northern fragment. So here is the route that we aspired to achieve. We want to unite this dipeptide with the fluorotryptophan that Dennis mentioned a few minutes ago, then couple in the phenylalanine, and finally unite that macrocyclic peptide to form the northern fragment. And this is an impossible synthetic route because there are way too many competing nucleophiles to be able to pull off all of these amide bond formations, but I've already given you the answer. We're going to be able to do this through the use of biocatalysis. And for the synthesis of the northern fragment, there are two enzymes that we're going to rely on. The first are ligase, or sometimes called ATP grasp ligases. And the way that these enzymes work is they take the carboxylic acid of an amino acid, convert it to an acyl phosphate. That's the form of activation. And then that activated amino acid is ligated to a peptide chain. Esterases, on the other hand, accept not amino acids, but amino esters or ester equivalents, and first convert them to an acyl enzyme intermediate, and then finally interception by a nucleophile forms the new amide bond. So let me show you one illustration of what protein engineering looks like. So for trip ligase, the natural starting point, the cat, to continue the analogy, was an enzyme that polymerizes tryptophan. Now, this isn't the function we needed to manufacture the northern fragment, but it was a starting point. And as we went round and around protein engineering, rounds of evolution, we were able to build new enzyme libraries where more and more of the desired function was revealed. And by round 7 of protein engineering, we were able to get activity on the desired substrates. One of the things I wanted to mention in innovation that was really important to enabling the use of ligases was on the energy side of the system. You know, ATP itself is extremely expensive to use as a reagent, and ATP powers ligases. And so in order to make this practical in a manufacturing setting, we needed to be able to recycle ATP. And actually, there aren't many examples industrially of ATP recycling, but Merck has been privileged to describe a few of them. The Islatravir science paper that I mentioned earlier describes one example of ATP recycling. It does require the synthesis of that acyl phosphate separately, and we were worried in the context of peptide manufacture that activated phosphate would acylate amines and other nucleophilic groups. Several years later, we published in the context of our manufacturing process for molnupiravir, our anti-COVID therapeutic, a different ATP recycling system that relied on the in situ generation of the activated phosphate species. This works really well, but the trade-off is a relatively complex system for recycling ATP. For Enlicitide, we turned to a different enzyme family called polyphosphate kinase, and this enzyme is able to shuttle back and forth phosphate that's from polyphosphate, sort of cheap source of phosphate, and ADP and AMP. So in situ can regenerate ATP from any ADP or AMP generated by the reaction. So returning back to this scheme, we now have a kinase that can recycle the ADP back to the active form, ATP, for the ligase processes. Each of these enzymes engineered through directed evolution for selectivity, activity, substrate concentration, the presence of inorganic phosphate, and many more. After 21 rounds of evolution, trip ligase is able to convert dipeptide to tripeptide, essentially perfect conversion and selectivity. Phi ligase, similarly, after 28 rounds of evolution, and macrocyclase converts tetrapeptide to northern fragment in perfect conversion, perfect selectivity. One incredible thing to note is that both trip ligase and Phi ligase come from the same parent enzyme, and protein engineering was able to divert two different evolutionary pathways to come up with final enzymes that are exquisitely selective only for their desired substrates and for there to be no crosstalk between trip ligase and Phi ligase. I mentioned that one of the benefits of using enzymes to perform chemical reactions is that oftentimes multiple enzymes can work together just like they do in every living thing to perform different reactions at the same time. And so we don't run this northern fragment process as multiple steps. We run it as a single cascade that combines these three building blocks into the northern fragment in a single step. This is a 4-enzyme cascade. There are no protecting groups used, no intermediate isolation. We use our new ATP recycling system to power the ligases. There's no high dilution for the macrocyclization to make the macrocyclic peptide, and we simply isolate the northern fragment via crystallization. We regularly perform this process at over 0.5 metric ton scale at our commercial facility. So that's step one. The eastern fragment comes through chemical synthesis from the building blocks I described earlier. So let's talk about how we put together the northern and eastern fragments to make diamine. We want to start by combining the amine of the methyltyrosine with that isopropyl ester of the northern fragment. But again, we have the challenge of too many competing nucleophiles to pull off this route. We're going to turn to a new enzyme family called the thioesterases. Now, for those of you who are less familiar with this topic, there's a family of microorganisms that make their peptides not through ribosomal chemistry like you and I do, but through non-ribosomal peptide synthetases, which are giant protein superstructures that, sort of like an assembly line, build peptides one amino acid at a time through the different interactions between the modules on the superstructure. So what we're really interested in is that final domain, the sort of terminating domain thioesterase that performs in natural species, either macrocyclizations or cleavage operations. But we don't want to add the whole NRPS superstructure to a vessel. We want to use just that domain. So we will need to excise it from its natural resting place and make sure it can be active separately to perform the chemistry we need. So in a schematic version of that, we have the thioesterase domain that we want. It has the natural reactivity we think we need. It's connected to a peptidyl carrier protein that then is connected to the rest of the NRPS system. When we express it without that peptidyl carrier protein, we expose to solvent a bunch of the protein that is normally not solvent exposed, and so it becomes very disordered. And we did a lot of protein engineering first just to stabilize this excised domain so that it could perform its enzymatic function. It's kind of like we went into an automobile manufacturing plant, which has a line of robots that perform different functions, and we decided we want just one of those robots. We took it outside of the plant to where we needed to use it, repaired it so it worked, provided the electricity and the utilities it needs, and now we're off to the races to engineer it for the rest of the performance criteria we need. So here's another example of what that substrate walk from cat to unicorn looks like. In the first round of evolution, we saw actually pretty selective reactivity for the wrong side of the eastern fragment, the benzyl alcohol and not the amine portion. So we actually took a step back in terms of substrate selectivity and blocked that benzyl alcohol as an azide to direct enzymatic reactivity to the amine. By the eighth round of evolution, we were able to see the desired site selectivity without the need to block the alcohol. And by around 10, we had desired reactivity on the actual substrates. But we weren't done yet. This is about 0.9% yield, very dilute, 2,000 weight percent enzyme, and a pretty unstable protein. In the end, it took 34 rounds of evolution, representing one quadrillion-fold improvement over our starting point to achieve our performance criteria. And I want to call out that it is essential to have ultra precise and accurate analytics along the way to be able to develop this kind of unnatural function. I also want to take a moment just to highlight that number, 1 quadrillion fold. When I was a PhD student, I worked on methodology with Barry Trost, and I suppose in the best version of my PhD, I could have taken a reaction that worked at 1% yield and made it work at 100% yield. And I never did that. But even if I had, that would have represented a 100-fold improvement. 1 quadrillion fold. I don't think it's any exaggeration to say that that kind of leverage on performance is the basis of life. The second bond that we needed to make to form diamine was between that alcohol and the amine on the fluorotryptophan. We were turned to 2 new enzyme classes to do this. The first is the ketoreductase that is able to convert ketones to alcohols and back. And the second is an imine reductase, which converts imines to amines and back, depending on how you run the reaction. And I've deliberately color-coded the cofactors suggestively because the fact that both enzyme classes can use NAD plus or NADH gave us the idea to perform this as an overall redox neutral transformation where we're just shuttling electrons back and forth, basically a biocatalytic version of a reductive amination. And this works really well. We were able to demonstrate this kind of process. And it's pretty remarkable, by the way, that both an oxidation and a reduction are happening simultaneously here. And because of the compatibility of enzymes, the ketoreductase and the imine reductase don't interact with each other. They only accept and deliver electrons to the substrate we designed them for and not each other. This is overall a hydrogen borrowing cascade, redox neutral with simultaneous oxidation and reduction. Cofactors are being recycled between these 2 enzymes, but unfortunately, this system is in equilibrium, and it became challenging to overcome the thermodynamic limit. So ultimately, we were capped in how much starting material we could push all the way to product. So we turned to protein engineering for a different purpose, which was not to increase the selectivity of the enzymes, but actually to change their cofactor selectivity. So if you forgive me for one more time, this car analogy, it's as if we had two gas combustion engines and we decided to change one of the cars to an electric car. And now those two can operate independently. So we use protein engineering to change the selectivity of the imine reductase, not for NADH, but NADPH. And now we had two orthogonal redox cycles with their own terminal oxidants and reductants that allows us to independently oxidize and reduce the relevant substrates and overcome the thermodynamic limit. Ultimately, we can run this process to achieve greater than 90% assay yield of diamine with perfect selectivity. Like in the northern cascade, we don't have to run this process as a series of enzymatic reactions, but can run it as an enzymatic cascade. In this case, it's a 5-enzyme cascade, no protecting groups, no intermediate isolation, our dual recycling system, selective macrocyclization to make diamine without high dilution, and again, crystallization is the way that we isolate diamine. We regularly run this process on over 400 kilo scale at our commercial facility. Okay, that's step 2. Western fragment comes from the chemical synthesis of the building blocks I showed earlier. So let's talk about the final step, uniting diamine with our Western fragment. So if you look at diamine, there are 2 reactive amines, and they're pretty differentiated sterically and electronically. And so when you run a chemical coupling of the Western fragment and diamine, what you can see is 2 different products. You see the desired coupling at the secondary amine and the undesired amidation at the primary amine. And perhaps unsurprising based on first principles, the undesired is by far the preferred mode of reactivity because the primary amine is much more reactive than the secondary. But that substrate control gave us the idea that maybe if we were able to temporarily mask that primary amine by first reacting it with a blocking group, we might be able to enable selective reaction at the diamine, the secondary amine, with the Western fragment. So here's how that worked out in practice. When we add no aldehyde, we see, as you saw on the previous slide, selective reactivity for the wrong amine. But if you add benzaldehyde first, which temporarily forms the respective imine with that primary amine, you see a remarkable turnover in selectivity to the desired product. Now, that chiral center next door that I've highlighted in blue is a bit of a challenge because once you've activated that amine, you start to see a polymerization there. So if you increase the electron density just a little bit of the aldehyde and use salsa-aldehyde instead of benzaldehyde, you're able to get the same regioselectivity that we see with benzaldehyde, but turn down the stereochemical erosion to get high chiral purity of the product. Finally, we need to create the last macrocycle of enlicitide, and we're going to turn back to the thioesterase family. I show here some examples of natural products that are made via the penicillin binding protein type thioesterase. And if you sort of squint at the structures down below, you can see they kind of look like the macrocycle, the final macrocycle of enlicitide. And that's one reason why we felt they were good evolutionary starting points for directed evolution. And I'm going to show the structure of the final enzyme API macrocyclase, and inside this pocket, sort of hugged by the enzyme, is the final intermediate of enlicitide ready to undergo final macrocyclization. After 26 rounds of evolution, we're able to achieve perfect conversion and selectivity through 76 mutations of that natural enzyme to form enlicitide. And like the other 2 steps, this is also run as a cascade, no protecting groups, selective macrocyclization. Actually, this process is run at simply at 10 volumes, which is, if you're a process chemist, you'll know is a very concentrated uh, uh, reaction volume. And again, Enlicitide isolated simply by crystallization. First is the subarate, and then in our pure step to decanoate. And we regularly run this process on over 400 kilo scale at our commercial facility. One of the jokes from the very beginning of this project have been that we would know we would have achieved our aspirations for the manufacture of Enlicitide if we were able to fit the chem draw on one slide, which we were not able to do for many, many years. But I'm proud to share this is that slide. I showed this bar graph earlier. Our aspiration to achieve commercial viability was going to require breakthrough innovation in chemistry and biology. And I'm so proud to share that the innovations described in the flash talk today represent another 90% reduction in cost for our commercial process. Ultimately, these innovations have enabled the discovery, development, and we believe the commercialization of Enlicitide to patients worldwide. The biocatalysis group at Merck is over 25 years old, and the first decade, I think, contained a lot of sort of exploratory work to try to understand if this crazy idea was even possible. Could you use recombinant enzymes, modified through protein engineering, these sort of white powders that, no organic chemist at the time was very familiar with? Could you actually use them to perform useful chemical transformations? And if you could do that, could you actually industrialize biocatalysis? And one of the papers that I'm really proud of, that sort of landmark entry into biocatalysis for the industry, was our publication of the transaminase that we use to manufacture sitagliptin, back in 2010. We've been very busy since those early days, and I highlighted islatravir earlier, but we've been able to apply cascade biocatalysis, so more than one enzyme at a time, to other important molecules at Merck. And today, the kind of problems that we're working on include solutions like cascade biocatalysis for peptides. Dennis asked a few minutes ago about sort of exciting areas for drug discovery, and there are so many other types of molecules out there that are being discovered that have really incredible potential for patients. And I think at the same time, we are only just getting started in the kinds of molecules for which biocatalysis provides unambiguous advantage for commercial manufacture and patient access. I want to close by returning back to that Saturday in November when I was, walking with my daughters to Starbucks, and I saw the push alert on my phone. We released a lot of data from our phase 3 studies that November, and I want to summarize our excitement with just a single graph. So, in one of our phase 3 studies, we had about 3,000 participants. About 2,000 of them got Enlicitide, about 1,000 of them got placebo. And everybody in the study is on stable lipid-lowering therapies, including at least a moderate or high-intensity statin, or have some documented statin intolerance. Okay, after 24 weeks, we asked the question, how many participants in each cohort are achieving their guideline-recommended cholesterol level of less than 70 mg per deciliter? And in the Enlicitide participant pool, so the 2,000 or so patient participants, 70.8% are able to achieve that goal. In the placebo arm, the answer is 1%. And the difference between those two bars in this graph represents our excitement and our hope for the potential of Enlicitide to meaningfully improve patients' lives around the world. When I started at Merck, the world in the pharmaceutical industry was neatly divided, in my perspective, between small and large molecules. But I think what molecules like Enlicitide and others like Enlicitide show is that there's plenty of room in the middle, molecules that aren't simply small molecules, aren't simply large molecules, that are able to take advantage of the best properties of both and drive really meaningful impact for patients. And in Enlicitide in particular, we're so excited to be able to deliver antibody-like efficacy and safety in the convenience and accessibility of a pill. So I mentioned at the start, I'm so proud and privileged to be here today on behalf of many, many colleagues at Merck and our partners who've contributed to this story over the last decade. Thank you again, Dennis and the Drug Hunter team, for the opportunity to be here this morning with all of you and to share the story with your audience. And with that, I'll be happy to take any questions.
Awesome. Yeah. Thank you so much, David, for like what really was a tour de force, I guess, and an achievement on so many different levels, like synthetically, but also, as you pointed out, the results from the clinical trial are very, very striking. So yeah, thank you so much for sharing this
Thank you.
incredibly scientifically rich story with us today. We do have a number of questions. I want to get started with one question. You mentioned that 1 quadrillion-fold improvement, and I really enjoyed how you how you talked about this. As we can only achieve 100-fold improvement if we go from 1 to 100%. I did notice, of course, the graph was a trillion-fold improvement, and in the first kind of 25 rounds, you barely saw anything. But I'm sure if you zoomed in, you did see small improvements. But I wanted to ask, like from like step 1 to step 2 to step 3, if like you don't see much improvements, like what are the things that you're typically changing in the next step to achieve better improvements?
Yeah, and that's a common question because you're right. There was a long period of time where we weren't even generating enough molecules of product to see it by HPLC. A lot of the really early work is just looking for hints in the mass spec that something was there. You know, at the beginning, it's not often the case that you have any reactivity on the substrate that you need, and it's not always obvious where you should start. The way that I told this story for thioesterase and the way that and other enzymes are described in the science paper are, skip over quite a bit of work that was done to explore different types of reactivity. And it's certainly true that we find ourselves chasing, different substrates, hoping that a little bit of reactivity on something that, if you kind of squint, looks like what you want it to be, will actually translate into the desired substrate. That's not always the case, and oftentimes we're looking at lots of different substrates to try to see hints of reactivity. But if you went and, for example, I've heard like Frances Arnold talk about protein engineering. What she would say is there's a universe of difference between 0% and 1%. As long as you see a little bit, that's the real miracle of life, of evolution, that you just need a little bit of activity. And if you can screen for it and select for it, eventually the exponential growth will kick in, and you'll be able to achieve the performance you need.
Okay, so you need those analytics and the perseverance.
Right. If there were 0 molecules in the screen of product, then you don't know where you are. But as long as there's a little bit,
Yeah,
life finds a way.
makes total sense. Yeah, super. Thank you so much. We have a number of questions from the audience. Let's start with Martin Fox. So Martin is asking, with the selective coupling of the diamine fragment to the Western fragment, the imine protection that you showed with the aldehyde is great. But was there a biocatalytic method for that where you could have,
Of course there was. Of course there was. So that disconnection is extremely challenging to do biocatalytically because it's a secondary amine, not a primary amine. And we worked for years on an enzyme to perform that coupling. And I don't want to get ahead of papers that have been written about that work. I'll just say, stay tuned. Yes, that's one that got away from us, and I'm grateful the team was able to come up with a creative workaround that enabled the manufacturing process. But yes, we worked for years on that.
Super cool. Yeah, next question comes from Julian Segert. They're wondering, what is the improvement on overall yield from the very first synthesis that was 70 plus steps to this manufacturing one? You mentioned the 290% reductions in cost.
I mean, I talk a little bit about that, or we talked a little bit about that in the paper that was published last year in the Journal of the American Chemical Society. I mean, many, many, many fold. I think like the real number is sort of unbelievable. Maybe the more important, or maybe the better answer to that question is, before we did all of this work, it wasn't possible. There was no yield, and with all of the cumulative innovation that this work represents, this is now possible. This is a viable medicine uh, for us to commercialize.
Totally.
Yeah.
Yeah, yeah. Andrew Becker is saying, "Hey, David, beautiful talk and loved all the analogies." I totally agree with that. You presented a very crisp story, but as you said in the intro, you tried a subset of the, over 3 million routes that
Yeah.
they're there. Um, what was the biggest strategic dead end in the process development that you ended up abandoning?
So the route as it exists now um, follows the same kind of retrosynthetic logic that goes all the way back to E.J. Corey and Woodward. Like the disconnection at the ring junction points provides the greatest overall simplification. So that in many ways is not new. I think it's very logical for synthetic chemists to do the disconnections there. I think what was less obvious was how to disconnect at the bottom of the molecule that where the eastern fragment and the what we call the linker of the northern fragment come together. And so that there was a lot of effort at the beginning to decide, if these were roughly the ring junctures that we would probably need to disconnect, what was the right order? And one of the main things that guided the strategy after doing a lot of experimentation was the ammonium containing side chain is quite tricky because it's charged. It's going to always bring along some counter ion. And so putting that in at the end, which is also how the clinical supply route works, I think anchored that as the end game. And the northern fragment itself is for like logical macrocyclic peptide of its own. So then it was how to start with the northern fragment and end with the western fragment. What is the order of the bond disconnections there? And so obviously the ones that I didn't show were ones that we looked at and ultimately decided weren't superior to the solution we ended up with.
Makes sense. Yeah, super cool. Along those lines
Yeah.
with kind of the ammonium, we have a question about the recrystallization process. How was the recrystallization carried out, and could you explain a little bit more about the process? Maybe that's also like after each step.
Uh, I think that it's a really important question, and I'm glad that someone's asked because the way that I just told the story and the way that it's described in the manuscript that was published in Science focuses rightly a lot on the biocatalytic innovations, but actually just as important were the crystallizations. If you look at impact to overall not just processability, but even viability. So it's a hugely important thing. And it's also something that I think gets less recognition generally from chemists. Like we're good at looking at, reactions and, synthetic routes, and maybe don't give as much attention to the importance of really good isolations, really good crystallizations. So there was a lot of screening that was done to identify crystalline forms. But one of the things that I'd like to highlight as a general matter, and then there'll be more papers that describe this more specifically. But one of the benefits that I described to using biocatalysis is that we were able to use a lot of unprotected amino acids, unprotected fragments, and having unprotected peptide fragments provided good handles for for salt formation. Like you saw the diamines, the bis HCl salt. There are other salts, the oxalate salt of the northern fragment, which you may not have been able to make if it was all protected with FMOC groups or Bach groups. And actually, those crystallization handles from the unprotected fragments, I think, provided really good toeholds to developing robust isolations of each of these intermediates.
Sounds good. Cool. Daniel is asking, so great talk, David. Around the time of the Istralvir work, it's Daniel's understanding that Merck was working pretty heavily with codexis to evolve the necessary enzymes through a saturation mutagenesis approach. And with multiple highly successful economic cascades developed here on the enlicitide synthesis by your team, how much of this process has Merck now internalized? And can you say anything about how you've improved the process development since?
Uh, we have done a tremendous amount of work to build up our internal expertise on sort of the whole pipeline of biocatalysis, from enzyme discovery and cell banking to protein engineering and fermentation. And I don't want to get too ahead of some of the papers that are going to come out to describe the work in more detail. I'll just say, stay tuned. We have a lot to share about sort of the next level down in the work that was necessary to pull off, some of the work that I described that kind of just feels like, and then a miracle happened and we had an enzyme. Rest assured, there's a lot of important work that happened to enable those evolution campaigns.
Makes sense. Yeah. Next question is from James Welts. Thanks for the great talk. Any enzyme classes or approaches that are limiting or too difficult today? So ATP costs and recycling used to be intractable, and decades ago, people thought that transaminases and PLP-dependent enzymes were difficult. What do you think is next?
I love this question. So I'll put a couple of the ones on my dartboard, that we haven't solved yet. For as a general challenge to the community, not just here at Merck. Halogenation can be very challenging, especially fluorination, and it's very challenging chemically for those of you who've made fluorine-containing compounds. And I know, speaking to a drug hunter audience, there are plenty of you fluorination people out there. They're hard to make. The reagents you have to use to make them chemically are not great either. It would be great if we had some better biocatalysis solutions to fluorination as well. I'll give you another functional group that sticks in my craw, Dennis, which is cyclopropanes. Again, not so easy to make chemically either. Not a lot of great biocatalysis solutions yet, but something that I'm looking forward to the field advancing into in the years to come.
That sounds fun. Yeah, I'm bow ties and
Right,
cyclopropyl
right.
groups. That's certainly all my jam. I guess in the interest of time, maybe we'll finish with one last question
Sure.
from Lucas. Uh, I would like to ask. So Lucas would like to ask your perspective on the growing role of peptide therapeutics in medicinal chemistry. How do you see the field evolving compared with traditional small molecules regarding the discovery, manufacturability, oral delivery, and production cost, in the years to come, essentially, with this groundbreaking achievement that your team now has?
I mean, I don't think I need to tell anybody on the call about two of the most important and probably largest drugs in the history of our industry are peptides now, and I think they, along with many other drugs, demonstrate how important this class of molecules is. But I will say, to tie this back to some of the manufacturing stuff we talked about this morning, solid phase peptide synthesis, dates back to the 1950s. It won the Nobel Prize in '84 for Merrifield. And the way that we've made peptides hasn't changed a lot in the intervening, 70 years. But I'm really encouraged, not just by work at Merck, of course, but other people, other companies in the field who are investing in new and better ways to make peptides, to make them more sustainable, more scalable, and ultimately to make the medicines more accessible to patients, because it's clear we can't keep making peptides the way that we have if the global demand for them is going to be this high.
Only. Do you actually see a lot of the methodologies also applied earlier in discovery? So like, enzyme evolution, is that something that people now apply earlier in medchem rather than just in process, or is that not yet trickling through?
Yeah, it's again not just Merck, but other companies applying biocatalysis to discovery. One of the challenges that protein engineering is still a pretty laborious exercise. It takes time, and if there's anything I've learned about my discovery friends at Merck is that they're very fast, and you need to be able to deliver the solution yesterday for the molecule they want to make. So like we need to be a bit more choosy about the kinds of problems we go after in discovery to make sure we have the solutions available when the team needs them. But yes, biocatalysis is also a really important enabling tool for drug discovery.
Well, with that, that was a very nice closing statement, David. Thank you so much again for an amazing, very clear, and also nice, like analogies in this presentation. Wonderful presentation. Really enjoyed it.
Thank you, Dennis. And thank you to everybody who was able to call in as well. It's been great to be here.
Yeah, thank you very much for the engagement of the audience as well. We had a couple of open questions that unfortunately, due to time reasons, we didn't get to. But yeah, we're very grateful for your engagement. And yeah, we will be back here on June 25th with a talk about the next generation utropin modulators for DMD. So that'll be by Angela Russell. So we look forward to seeing you then. And until then, I wish everybody a wonderful rest of their day and a wonderful weekend to come. And thank you again, David. Bye, everyone.
Bye.